In this video, we're still talking about chapter 1, but we're going to begin by talking about benign and malignant tumors. So when we talk about the development of cancer, which is oftentimes referred to as tumorigenesis, there are often some kind of event that occurs on the cell, and it's going to cause it to start to change over time. And when we think about regular cell processes, we know that the cell has its own metabolism and it's going to be going about its normal day. And sometimes the cell might increase or decrease its size. In general, the size can kind of stay the same within a tissue specifically. But it has the ability to increase its cell size, right? So if we were to look at cells, we could see that there's an increase in cell size in certain cases, right? So if we were to observe that, we would call this hypertrophy. So we have not an increase in cell number. We still have the same number of cells. All the cells are organized in the same way. They're not acting funky or anything. The only thing we see is an increase in cell size. So this could be reversible. It could increase and then decrease again. There are certainly cases where they increase in cell size and then they don't change back. But this could be a normal response to what is the body responding to? Is there a stimulus that's causing this to happen? And certainly this could happen in response to exercise or something like that. In another case, we could have cells that actually increase in cell number more than what would typically happen. And this is what we call hyperplasia. And so some of our tissues might respond to various stimuli simply by increasing cell number. And then maybe later they'll slough those cells off and then you'll kind of return to a regular case, right? So this is potentially reversible and does not necessarily mean that you have cancer. So one example of that would be calluses, right? So that's a stimuli to, the pressures that are being on the foot or the hand or wherever it is. And then the skin will kind of increase the cell number in that area and offer the body some protection. Now, in this case, we're talking about dysplasia. So dysplasia, we also have an increase in cell number, but we have abnormal growth. And we see that they're not organized as well as they were before. So they're starting to act funky, right? So we have abnormal growth, we have disorganization, and we can have various degrees of how disorganized these cells are. So we could go from mild to moderate to severe dysplasia, right? So dysplasia can be considered on the path towards development of a tumor, but isn't quite a tumor yet. So, and in the world of like uterine cancer or cervical cancer, when pap smears are done and the tissue is examined, then this is where dysplasia could be picked up very early and kind of taken care of before it progresses into a tumor. The next one is neoplasia. So neoplasia, you would see an increase in the number of dividing cells and disorganized growth. So we have both of those happening, and you can see that it's all just a mess in here, right? So it's uncontrolled abnormal tissue growth. But neoplasia could be benign or it could be malignant. And when we talk about benign, it's not necessarily cancer yet. And it may never become cancer. It's just an abnormal tissue growth. But this neoplasia could also progress into a malignant cancer type. So some tumors are considered a neoplasia. Something to consider, we've been talking about cell number and cell organization. But another thing to consider is cell differentiation. So when we learned about cell differentiation in general biology, we were referring to what is the process that cells go through from a stem cell and then to where it's a cell is going down a path where it's going to become a certain type of cell, whether it's a fat cell, bone cell, blood cell, a nerve, anything like that, right? And so this required a programming of transcription factors that caused a cell to go down a certain path. And we talked about once it reached that path, and in general, that was kind of like a one-way street, right? Like once it became a muscle cell, it's going to stay a muscle cell. But cell differentiation is that really complex process that puts a cell down a path towards differentiation. When a cell is fully differentiated, it is its final cell type. It's a muscle cell. It's a fat cell, right? That would be a fully differentiated cell. And that cell would have its own special structural organization. It might have certain proteins or products that it produces. And in general, cell division and cell differentiation are kept in balance. So just because a cell has been differentiated may not mean that it's never going to divide again. Some of them do divide again and some of them do not, right? But that cell division and cell differentiation, it's all being kept in balance. An example of this will be looking at your skin cells and how the skin kind of continues to produce new layers of that epithelial layer, right? So there's a lot of balance that's happening in the system. So when you look at the skin, right, it's made up of basal cells and squamous cells. And basal cells in this image here, right, are going to be found in the epidermis. So you have a couple of different layers in this epidermis, right? So at the bottom of this layer, you have your basal cells. And you see it's just a single cell layer right here. And we call that the basal layer. And these cells are round, so they have kind of a distinct shape, but they do undergo cell division. And then one cell typically stays in the basal layer, and the other one differentiates and moves up to the next layer and becomes a squamous cell. And the squamous cells are the light pink cells here. And you can see there's multiple layers of them on top of the basal cells. So the squamous cells would make up the surface of the skin. There's going to be multiple layers. And these cells can be thin and flat. They can look like fish scales, but they protect the body, right, from friction, mechanical damage, and from the outside world. Some of these cells might be keratin, right? And then when these cells kind of are up at the top layer, and then you can see that they can kind of die, and then they'll kind of make that top layer and slough off, right? But you can see how they're going to be constantly replenished from the basal cells underneath. So if we look at the normal growth of this epidermal layer, we have our basal layer. So these are our dividing cells. And we have our squamous cells in this kind of tan up here. And as the new cells are made, they're going to get pushed up to the next layer of squamous cells. At the top of all these layers, we have some dead cells. So they kind of reach the top and they start to die, and then they're sloughed off, right? They're shedding. But the new squamous cells kind of reach that top layer and replenish it, right? So it's all moving in this upward direction. And if you kind of take this down below and look at, okay, here's one basal cell. It divides into two cells, right? So one of them becomes a squamous cell and the other one continues to be a basal cell. But it's always a 1-to-2 ratio. So 1-to-2, 1-to-2. And these squamous cells are going to keep moving up through the layers and eventually go away. So they do not accumulate, right? Now let's look at tumor growth within the epidermal layer, right? So we have our basal cells at the bottom and our squamous cells again right here. But what's happening here? We have some abnormal cell growth here. So in this case, so here we have the basal cell here. It produces a new cell that goes to the squamous layer, and then the other one stays in the basal layer. But let's say that we have a basal cell that goes kind of wonky and it reproduces and makes 2 cells. And then this one reproduces and produces 2 more. And this one reproduces and produces this one. And so it's going out of, it's just going out of whack. It's producing too many cells for every basal cell. So instead of a 1-to-2 ratio, now we have a 1 to 1, 2, 3, 4, 5, 6, 7 ratio. So we have this buildup of these cells, which is what contributes us to developing a tumor. And you can see that some of these tumor cells are still basal cell, and some of them are squamous cell, indicated by the different colors. So it's a heterogeneous population. Some of them are undergoing differentiation, while some of them are not. And in this case, if it's not differentiating, then that basal cell is going to continue to make new cells and continue to contribute to this tumor growth. So when we talk about cancer, we're talking about, and tumorigenesis, like what is the process that causes normal cells to go from this normal organized situation to where they start to become like moderately differentiated and then severely undifferentiated. And once and it's a process, it doesn't all happen in one go. So there's going to be multiple steps to go from here to here and then from here to here. And so what are the causes of those steps? What happens at the gene level, the RNA level, the protein level? What causes all of this to happen? And so that's all part of studying cancer biology, right? So cell differentiation is a very fine balance under normal conditions that will keep everything the way it needs to be. But once the balance becomes tipped to where things are not differentiated or things are dividing too fast or dividing too slow, then that's where we start to get in trouble and where a tumor could start to grow. And here's a picture from the book showing colon cancer. So on the left are some normal cells in colon tissue. And so again, there's going to be like a couple layers of cells within the colon. So this would be like the surface layer of the colon that's facing the lumen. And it's going to have some globular cells that are going to be lined up here. And here you have some cells that have kind of a column shape, right? So these are kind of normal. But as you look at this section, you see a lot of disorganization over here. You see cells stacking on top of each other. And here you can really see that everything is just not the way it should be. So you don't see your distinct layers and you're losing your organization here, right? So this is indicative of colon cancer. So you're going to come across these terms when we're talking about cancer. You're going to come across benign. So a benign tumor, I mean, a tumor is going to be a growth of cells, an abnormal growth of cells. But if it's benign, it's going to be confined to its local area. And it may not be life-threatening even. Some of the benign tumors can be taken care of surgically or by other methods. And the survival rate for that individual could be pretty good. However, if you have a tumor that is malignant, that means it's invading the surrounding tissue. It could enter the bloodstream and then spread to other parts of the body. And that process is what we call metastasis. Then that can be a very, very dangerous situation. And that's the survival for malignant tumors is not very good. And then the term cancer is a generic term. It could refer to any malignant tumor or any tumor that is capable of spreading by invasion or metastasis. It doesn't mean that they have invaded or metastasized yet. So we're going to go through a couple of the properties and kind of comparing benign versus malignant. So in this table, we're comparing benign and malignant. So for a benign tumor, the growth pattern is going to be localized. A malignant, it's going to be spreading out. It's going to be invading and going to other parts of the body. So a benign tumor is rarely life-threatening, but a malignant tumor is often. And then growth rate, benign tumor is usually slow, And there are some fast growing benign tumors, but it's not typically the case. And the malignant tumors can be very rapid. State of differentiated, a benign tumor still is pretty differentiated, but a malignant tumor typically is not, but it can be variable. So you'll see a lot of like kind of vague descriptions, but that's because there's such a wide variety of tumors and every tumor has its own characteristics. So you could have a malignant tumor that still has some differentiation, but you could also have one that's very, it has lost all of the differentiation. And so that's kind of what this chapter goes back and forth, is ways in which some of these different cancers are different. And then we'll talk about the naming of these cancers. So you'll hear the terms carcinoma and sarcoma. And so they indicate the origin of the tumor. So a carcinoma would arise from the epithelium. So this could be a covering of an external or an internal body surface. Carcinomas are very common and they do metastasize. They can spread through the lymphatic system. And some examples would be lung, breast, and colon carcinomas. Sarcomas, on the other hand, arise from connective or muscle tissue. So they could come from bone, cartilage, blood vessels, fat, fibrous tissue, and muscle. So these are more rare, but they can metastasize and typically would do so to the lung. And looks like I need to finish filling this out, but you could think of like osteosarcoma, which is a cancer of the bone. So any kind of cancer that would arise from these various tissues. There's two other groups. So we talked about carcinoma and sarcoma. The other two groups are lymphoma. So these arise from lymphocytes, which are white blood cells, and they will mostly grow as a solid mass of tissue. Leukemia is blood origin, and this would be proliferating within the bloodstream. So there wouldn't be like a mass. There's just a larger number of those blood cells within the bloodstream. So those are the four major groups of tumor types. Naming cancers usually has a couple of different rules. So there's usually a cancer, like a prefix that's going to indicate the location or the origin of the type of cell that's involved in this cancer. And so here's an image showing the prefix and matching up to the meaning, right, that's next to it. So you can kind of look at this, right? Hepato would be liver, right? Lipo, like lipoma, or is fat, right? So a lot of things that you probably recognize here. And there's lots of resources online as well. I put an example here of a website that, I don't know if it's listed here, but it is here at cancer.gov. So just realize, and I have the link here, you can click here and take a look at this website. Any one of these things that's in blue, you could click on it and kind of get some more information. But it would take you through the different tissue types. And then within that tissue type, what are the various kinds of cancers that are there and what are their names, right? So, I mean, basically a cancer can arise from any cell or tissue within the body. So there's many kinds of cancer, right? And then depending on what kind of cell type or where the origin of that cancer arose from, then these different cancers will behave and respond to treatments differently. So therefore, it is important to correctly classify a tumor by its origin so that you have a direct vision for where the treatment plan is going to go. And you have, like the past data to kind of show you what is your best opportunity for responding to a treatment, depending on what kind of cancer you have. And we talked about cancer can arise from any kind of cell type or tissue type, but also keep in mind that, even if we're talking about one tissue or one organ, that there could be different kinds of cancers that arise from there. So that adds to the complexity. So in this example, we're looking at lung cancer. And you can see in the pie chart that there's within the category lung cancer, there's lots of different types. So, and they're in general broken up into small cell lung cancer, non-small cell lung cancer. But then there are some others that don't fit into those classifications, right? And you can see that there are going to be specific kinds of cancers that will arise from different areas within the lung, and the different cells that make up the lung. And so it's important to classify it correctly because they're going to have different responses to treatments. And we had talked about the fact that cancers can arise in one location and they can metastasize and move to another location in the body. But a cancer will be classified and named and discussed with regards to the name of the primary tumor. So when we say primary tumor, we're talking about where did that cancer arise in the body first, even if it arose, say in the, even if it arose, in this case, it's talking about, a liver cancer, and then it has metastasized to the lung. So you do have cancer in the lung, but you would refer to this cancer as a liver tumor with metastasis in the lung. So this is the original tumor. It arose from these kinds of cells, and it is those cells that move through the body and moved to the lung here, right? When we talk about the different cancer types, we often talk about 5-year survival rate. So this refers to what is the percent cancer patients who are still alive 5 years after initial diagnosis. And so they're going to be collecting that information for thousands and thousands of patients, right? And the number that's presented is from the time of diagnosis, looking at 5 years later, how many of those patients are alive. And so we often kind of talk about what is the survival rate for various tumors. It could be 5 years or it could be 10-year survival rates, but a lot of times we're talking about 5-year survival rates. And even if a patient was alive at 5 years and died in the 6th year, they would still be counted in this statistic, right? And you can see that, different tumor types have different 5-year survival rates, right? And they can vary quite widely, right? And so that's kind of shown here, the different tumor types, and there's 5-year survival rate. Something to keep in mind is lead time bias. And so that's kind of, and it's shown here graphically that here's the usual practice, right? So you have a person who has their onset of their disease, but they don't know it yet, right? And usually by the time the disease is detected based off of symptoms, then it's quite a bit later in the progression of the tumor. And then if you kind of followed them over time and you looked at when they died, you would say this was their perceived survival time from the time of diagnosis to the time of death. Now, with advances of screening and detection measures, instead of detecting the disease at the onset of symptoms, you might actually detect them quite a bit earlier when they don't have any symptoms yet. And so your perception of their survival rate is going to look much, much larger, right? So you're going to look at that and say, look at this survival time. It's huge, right? So when we look at some of this data, we have to keep this in mind, that sometimes there's this lead time bias that gives us the feeling that some of the practices or measures we're making are actually increasing survival time. But if we look at this graph here, there's actually no increase in survival time. It's exactly the same. But let's say you graphed it out and you said, well, this person had a survival time here, and we could say like 10 years, but this person had a survival time of 30 years, then we would feel like we were successful, right? So always keep that in mind when you're looking at the data, that there could be some kind of bias or something kind of underneath that you haven't quite observed that's causing you to think that the data looks way better than it does. Okay, so going back to our benign and malignant tumors and kind of looking at what is the difference between them. So when we're diagnosing these tumors, one of the first steps would be microscopic examination of a biopsy, and then kind of getting to the point where can we classify the tumor, can we stage it, and kind of determine where we're at from the beginning. So when you're looking at the microscopic appearance of these tumors, here are some of the things that you would be looking at. So a benign tumor would have small nuclear size versus malignant, which is large. The volume of the cell, the nuclear size to cytoplasmic volume, will be low in benign, high in malignant. The nuclear shape will be regular for benign, but irregular for malignant. Mitotic index, so it's the relative number of dividing cells, is low for benign, high for malignant. Tissue organization is normal for benign, disorganized for malignant. Differentiation is going to be well differentiated for benign and poorly differentiated for malignant, and that's sometimes referred to as anaplastic. Tumor boundaries will be well defined and poorly defined for malignant. So examining under the microscope, a pathologist can look at the structure of the cells and get those various measures and determine, is this cell benign or is it malignant? And so when we talk about, like, the boundaries of these tumors, here it has well-defined boundaries. For a malignant tumor, it's exceeding those boundaries. The cells are starting to invade into the tissue or the area outside of where those cells would normally be. So mitotic index, we mentioned that before, has to deal with mitosis. So what we've talked about in general biology and cell bio. So there's various phases: interphase, prophase, metaphase, anaphase, telophase, right? So where you go from a cell that's one cell, it has duplicated its genome, and now it's splitting off into two cells, right? So mitotic index can tell us what is the rate of cell division happening here, right? So in looking at the cells in the tissue specimen, then the pathologist could take a look at, well, how many of these cells are in mitosis, and compare that to the total number of cells. And so by microscope, chromosomes are visible when the cell is undergoing mitosis. And then you can see the various stages of mitosis, right? So here they're aligned at the metaphase plate. Here they're pulling apart, right? And then you might even be able to see some of the cells starting to split, right? So you can kind of take a look at which one of these are in mitosis. And if they're in mitosis, they could be in any one of those phases, right? So someone could take a look at all the cells in the field of view and say, okay, the cells with the visible chromosomes are the ones that are in mitosis. So I count 20 of them. The cells without visible chromosomes are not in mitosis, right? Because when a cell is not in mitosis, the chromosome structure is relaxed, and it's not bundled up into these tight chromosomes, right? It's called chromatin, right? Like it's loosely kind of more relaxed. So these cells are not undergoing mitosis. So if I do the division, cells in mitosis over the total number of cells, then I can get a ratio. So this is a mitotic index, right? 0.267. So you could get a mitotic index, and that'll give you an indication of, like what is the rate of cell division that's happening in this population of cells. So after all of this examination, then a pathologist could begin to do some tumor grading. And tumor grade usually has grades 1, 2, 3, and 4, with grade 1 being lower grade, grade 4 is higher grade, and we call anaplastic. So that was like that very disorganized, not differentiated kind of cell, right? So we have well differentiated on the low grade end, moderately poor anaplastic. So as the grade gets higher and higher, then the tumor is more and more disorganized. And then we can move on to tumor staging. So tumor staging is a system that's used to classify the extent of the cancer. It kind of gives an indication of how large is the tumor, has it grown, has it spread. And so in many cancer types, the TNM system is used. There are some other systems out there, and the system is going to be specific for that tumor type, right? This will be based off of clinical, radiological, and surgical criteria. So the TNM system, so T stands for tumor, N for node, M for metastasis. So what is the tumor size? How deep is it? Has the tumor invaded into the local lymph nodes around the cancer? And so how many lymph nodes are involved? And then metastasis, has the cancer spread to other parts of the body? And putting those three measures together, so you're going to have a measure for T, a measure for N, and a measure for M. So based on that, then now you're ready to stage the cancer. So this is an example of bladder cancer. And so you can see that there's stages 0, 1, 2, 3, and 4. And each of one of those has a certain classification that would cause it to fit it within that stage, right? And so the more lymph nodes that are involved, and if it has metastasis, then that's going to cause it to be a more high grade tumor. So stage 4 obviously is very high grade tumor and very aggressive. And you can see that also in the image. It's pointing to, like, here's a T1 tumor, which is starting to invade into the local tissue. And then as it invades more and more and gets into the other layer, like the muscle layer outside of the bladder here, then the stage is going to be higher, right? And then you see T4 is when the tumor had invaded into nearby organs. So those are two different things, the grading versus the staging. So grading will tell you how abnormal are the cells, and it's on a scale of 1 to 4. It's based upon the microscopic examination, and it will look at different features of the cell, like the shape, the size, differentiation, and the mitotic rate. And it measures the likely aggressiveness of the cells. Staging is different from grading, as it discusses, or it's looking at how far the cancer has spread. And then you typically have 1, 2, 3, and 4 different stages. One of the staging systems is called TNM, but there are other systems out there. This would be based on clinical, radiographical, surgical examination, and it takes into account these various features, and it measures the levels of tumor spread throughout the body. So you can kind of see how these things are similar but different, right? And then what? Just looking at past tumors and the history of those tumors, the tumor grade and tumor stage are indicative of the future for that cancer patient, right? So if we look at graphs that look at 5-year survival for tumor grade, you can see that for tumor grade 1, the 5-year survival is really, really good. But as you increase in tumor grade, it decreases quite rapidly, right? So that's almost 50% decrease. Tumor stage as well. Tumor stage 1 is really good, but by the time you get to tumor stage 3, the 5-year survival rate's like below 30%, right? So it's very indicative of what is the future for this patient and what is their prognosis. So the very last part of the chapter is kind of an introductory overview, and it actually encompassed quite a few topics. which I'm not going to talk about all of those here, but I will kind of briefly talk about tumorigenesis. So tumorigenesis, like we talked about before, is the formation of a cancer. Some other terms that are kind of synonymous with this would be carcinogenesis, oncogenesis. And the tumorigenesis process is very complex. There's many different steps that happen on the path between a cell being a normal cell and then becoming a malignant cell that has lost their differentiation. So, and there's a lot of different things that can happen in between going from normal to malignant. A normal cell can start to become like on this pathway due to a number of different stimulus. So it could be a chemical, a radiation, a virus, a mutation, things like that can kind of cause this process to get started. And we call that step initiation. So once the initial damage has happened, now the cell's kind of initiated and then has the ability to go through this process. It doesn't mean that it always goes through that process, right? Within this process, there are a couple of terms, and we'll be talking about these a lot, oncogenes and tumor suppressors. And these two terms specifically talk about genes within the cell. And the gene could either be mutated or the expression of the gene could change such that it changes the direction of the path for that cell. So I kind of put this together to compare and contrast those two things. So an oncogene, and this refers to normal genes that are encoded in the genomic DNA and have normal functions. So they have a normal job within the cell. So an oncogene would be a normal gene that becomes mutated or overexpressed, meaning that the cell starts to make more and more and more RNA, which results in more and more protein and causes it to act differently than before, right? Because there's too much of it. But an oncogene, when overexpressed, can contribute to the development of cancer. So some mutations can alter a gene product that will cause it to increase their expression or cause it to be constantly turned on. And some of them cause an increase in cell division, right? So actually many of them lead to an increase in cell division. So once you have this activation of cell division, you have uncontrolled proliferation of a cell. And so that's one of the very first steps to developing a tumor is uncontrolled cell growth, right? This could be produced by a number of different ways. So it could be a point mutation, gene amplification, chromosomal translocation. The mutation will occur in somatic tissues, so it doesn't typically occur during embryonic growth. It typically occurs like within some somatic tissue. So oncogenes, once they've been turned on or mutated, typically result in more aggressive behavior of the tumor. Some examples of oncogenes include RAS, HER2, BCR-ABL, EGFR, and VEGF. And some examples of the cancers that arise from this would be chronic myeloid leukemia, CML, breast cancer, or kidney cancer. And tumor suppressors is another kind of class of genes. So again, they're normal genes that are within the body, right? And their job in the body is to protect against uncontrolled cell growth. But mutations occurring in those genes will lead to progression of the cell cycle and causing the development of tumors. So tumor suppressors are like the brakes for cell division. And the mutation that we're talking about is like an inactivating mutation. So it's almost like taking the brakes away. And so the cell cycle is going to continue to divide. So when you have the loss of the tumor suppressor, then you actually have a gain in cell division. So this can occur by a number of different ways. So it could be deletions or point mutations in the gene. There are examples of that occurring in somatic cells and the germline cells. So some of the germline cells, when that mutation is there, then you're going to see that mutation in many different tissue types in the body And you'll see there's some syndromes caused by this where tumors kind of arise in multiple areas throughout the body. So in this case, cancer development is typically recessive because typically just one of the alleles would be mutated. And so it's going to be a little bit less aggressive than with an oncogene, but still dangerous nonetheless, right? So some examples are RB, like the retinoblastoma protein, APC, and BRCA1. So BRCA1 is been shown to be involved in breast cancer, right? The retinoblastoma protein is involved in retinoblastoma. Your chapter kind of gets into a review of basic DNA kind of structure, function, and goes through transcription and translation. So I'm not going to go through that here, but I'm asking you to just review the basics of DNA and what are the bases A, T, G, and C? What's the base pairing that happens? And then just in general, the process of transcription and translation. These two processes are important to understand because the normal function of transcription is to make mRNA, translation is to make protein. And as long as the processes go right, then you have normal cell function. But if this process creates a mistake in the genome, then that's where we have issues arise, right? So that's where we could get genes with mutations, which could cause cancer in the end, right? So some of these concepts will come up in some of the later chapters. So that's it for this chapter. Thank you for listening.